away we go. Okay. So the question was about activated B cells and memory cells, and then first and second exposure events, which is a really good question for a variety of reasons. But a big one is it kind of gets at the cool, but also very important, and also conceptually a little bit tricky for some people, concept of what immune memory really is and how it works depending on whether or not we've been exposed to a particular pathogen before, or whether this is our first time experiencing it. So the question was about activated B cells. And so I'm going to kind of run through B cell sensitization and activation just really quickly up in this left-hand corner so that we cover all of our bases for how they get activated in the first place. And then we will look at the rest of it. So B cells, and remember, B stands for bone marrow because these cells become immunocompetent in the bone marrow. And they're a little bit different than T cells because they have antibodies on their surface, which are little Y-shaped guys. So that's criteria number one. But remember, we also have to consider major histocompatibility complex proteins, right? And so those are going to play a role as well. It's important to remember of the MHCs, we've got class 1 and class 2. Any cell with a nucleus has class 1. And certain cells that function in the role of antigen presenting cells and lymphocytes will also have class 2. And so I'm not going to draw the class 1 MHC on this B cell because it's not going to factor into what's going on here. I'm only going to draw the class 2 because that's the one that is involved in sensitization and activation. So I'm going to make it be orange, I guess. Okay, so the reason I'm making it shaped the way it's shaped is because these proteins are holders for things. So the class 2 MHC by itself just means this is where I hold things that I would like to show other immune cells. And it's where I hold things that I would like to show other immune cells without having them kill me because I don't want them to think I'm infected with something. So that's kind of the gist here. So let's do some labeling just because I don't want to be a naughty professor who doesn't label anything. Okay, so this is B cell. That is an antibody. And this is our class 2 MHC. And you'll notice it's currently empty. So let's say, for the sake of argument, that, and I'm going to be boring and make antigens red again because why not? This B cell encounters an antigen. And so because of that, maybe the antigen happens to stick to this antibody. And maybe another one sticks to this one. So that's exciting for the B cell because not everything sticks to the antibodies. So now what the B cell has to do is get the antigen into its class 2 MHC. So that is going to be essentially, and I'm not going to draw it because it would take too long, but remember endocytosis, where the cell can take things into itself by having a vesicle form and pinch inward? That's what happens. So there's a kind of endocytosis called receptor-mediated endocytosis or antibody-mediated endocytosis, which basically says that when something on the surface of the cell is bound to something else, when it catches something, you can draw the caught thing inside by endocytosis. So that's what the cell does. It's going to basically draw the antibody antigen complex inside, separate them, and then stick the antigen in a class 2 MHC and stick that in its membrane. So the net result of that will look like this. So I've got antibodies and our little class 2s. Now, both. So antigen in antibody and antigen in class 2 MHC. But we're not done yet. So this is the first step, and it's an important first step, but we still have to activate them fully. So these are what are considered to be sensitized, but they're not activated. So for activation, we need a CD4 helper T cell specifically. So fortunately, those aren't hard to find. Remember, I was talking about lymphatic tissues last night, if you were here for that. And I was just saying that the lymphocytes you see in the bloodstream are typically on their way to somewhere. The majority of your lymphocytes in your body live in lymphatic tissues situated all over the place. So lymph nodes, for example, are hotspots for T and B cells to live, and it makes it easy for the T and B cells to interact because they're already in close proximity to each other, which is handy. So it's not like this sensitized B cell has to go on an epic journey to go find the helper T cell of its dreams and become activated. Odds are the helper T cell is already nearby. So just an important sort of way for us to connect this process to a location inside of humans. Fun fact, what I'm about to describe to you is also the reason that when you are sick, your lymph nodes become swollen. So, okay, so we've got our sensitized B cell, and he needs to find a CD4 helper T cell, which I'm going to draw in a slightly different color scheme just to differentiate them. So here he is. Actually, I'm going to draw his receptor first, because if I don't, I'm going to maybe make him too far away. There we go. Okay, I feel good about that. So CD4 helper T cells are a special kind of helper T cell. They're the major coordinators. Specifically, they're very good at coordinating immune responses that involve both T cell mediated immunity, so cellular immunity, and B cell mediated or humoral immunity. So they form this link between the two halves of the lymphatic immune system, AKA the part of the immune system that can learn. So acquired immunity versus innate immunity. So we have a receptor, which again, I'm going to label. So it has to match the antigen, at least enough for them to bind. So it can't just be any old helper T cell. It's got to be one with the appropriate receptor. And then this is our and then I'm going to just add in our little CD4 protein, because that's what kind of distinguishes it. And so that looks like this. So once this docking step occurs, where the receptor binds to the antigen that's bound in the class 2 MHC of the B cell, that creates a scenario where recognition occurs. So I'm going to draw recognition as being, oh, let's do that in white actually. So recognition is aha. And then the next step is co-stimulation. And without going into cytokines too much, because there's lots and lots of cytokines and they have many functions and possibilities. Co-stimulation is illustrated in your book as just being an arrow, and that really throws a lot of students for a loop. The reason for that, once more, is that which chemicals are being released during co-stimulation are context and case dependent, and they are many, and they're complicated, and they're just kind of beyond the scope of this class. So instead of saying all that stuff, your book just says, hey, some chemicals are released by the cell, and that's co-stimulation, full stop. So this comes in the form of cytokines. But I'm not going to specify at this time which ones, because it's not important. Okay, so the net result of this is then we get, once this set of events has occurred, now our B cell is both sensitized and activated, so it's ready to go. So let me draw it. In all of its activated, sensitized glory, with all of the things and the stuff that it needs to have. So now what's going to happen to this is clonal selection, which is the rapid mitosis of this cell to produce many, many copies of this same now activated and sensitized B cell that are capable of doing stuff. So in the humoral immunity, what this looks like is you get one population that are plasma cells, and they make antibodies, which I'm abbreviating as alpha Bs because it's easier to write. So you see that, that's what I mean. And then also we've got memory B cells. And I want to stress about the plasma cells, the antibodies they make are going to be primarily antibodies that will be released from them and float on their own in the plasma. And I'm making that clear because I want to distinguish them between, or distinguish them from the antibodies that you would find on the surface of the cell. So these are going to be immunoglobulins that are like just floating in the plasma, which is great because then they can float in the plasma and in the extracellular fluid and stick to any antigens that might be present anywhere. So having them be free floating is an advantage. And then memory B cells, those do what it sounds like. So they're not going to necessarily participate in the battle that's happening against the pathogen right now. Instead, they stick around in case that antigen comes back later. So the question that was asked, which is a really good one, was, we know that memory B cells and plasma cells result from clonal selection of an activated and sensitized B cell. And then if this is a first infection, what we would see is a coordinated effort by the immune system to get rid of the infection. And so what you would also see is that, you know, infections end, right? The antibodies are going to bind to the antigens and they're going to be cleared out of the system. T cells are also going to be involved to kill anything that's been infected. You might even have a fever, et cetera. So once all those things have concluded the infectious period of the disease and you're successfully cleared your antigens out of your system, the memory cells are like, yeah, but it might come back. So I'm just going to keep this antibody that's specific to this thing here, and I'm going to remember. So the question was, of these memory B cells, if the antigen comes back a second time, and they initiate clonal selection once more, does the resulting population of memory cells differentiating into? So basically, if a memory B cell encounters the same pathogen again, it rapidly activates a group of plasma cells to pump out antibodies against that antigen immediately instead of having to wait for all this to happen. And the question was, does this secondary exposure event that results in plasma cells, does it also refresh the supply of memory cells? And the answer is yes, it does. And that makes sense, right? Because memory cells are there for remembering. And if you've had a recent exposure to something that you've encountered before, that would suggest that that pathogen is in the environment that you're in. And if it's in the environment that you're in, being able to remember it very well is probably a good idea, and it will provide you with a survival advantage. So for that reason, maybe just refresh our stock of memory cells if we have a re-exposure event. So what this does, among other things, and I'm going to draw a little chart up here, is essentially allows the body to respond much more quickly the second time a pathogen is encountered. And the difference is significant. So I'm just going to make this arrow shorter. And so obviously the plasma cells are going to rapidly create oodles and oodles of antibodies against that particular antigen with the goal of binding them up and getting rid of them, nipping that problem in the bud before it even has a chance to take hold. So this chart that I'm going to draw you is similar to one that's in your book, but I'm not going to include as much detail on mine because I'm just trying to make a point about a difference rather than focus on specific numbers. So if you're like, oh, that looks like that chart, but she's not doing it the same, that's why. So this is just going to be antibody titer, which means if you collect plasma and you titrate it and you count the antibodies in it, how many are there? Are there lots or are there not very many? So you can also think of it as just being amount. And then this axis is time, typically in days. So in a first exposure event, and so your book chart distinguishes between IgMs and IgAs for this, and I'm not going to. I'm just going to do kind of a basic rundown of this. So if it's a first exposure event, because the pathogen has to enter the body and then make more of itself or increase in number. So if it's a bacteria, maybe it divides and makes more copies. Or if it's a virus, maybe it gets into our cells and makes our cells make more copies. Regardless, there's going to be a period of time after you receive the infection that it hasn't really gotten underway fully yet. And so it's not been detected by the immune system yet. That time varies depending on the pathogen, but it's often between 2 and 4 days for an average incubation period. This is the time that pathogens really capitalize on to make us sick, because they are basically enjoying the freedom to operate relatively unnoticed by the immune system, which means that they have the power to do more damage because nothing's fighting them yet. So in a first exposure event, you might see starting at around day 4, when the immune system is like, oh, there's a bad guy, you see an increase in antibodies over the following few days. But the antibody response takes some time to mount, because you have to make a bunch of clones, and then the clones have to make antibodies, and so forth, so on and so forth. So this is first exposure. Now, in second exposure, because we have these memory cells and they're going to be sticking around to notice, if I get re-exposed to the same pathogen a second time, my antibody titer shoots up on day 1. So this window of time that the pathogen would have to, be nasty in my body and cause problems is not going to be permitted on the second exposure, because now the immune system is like, hey, I remember you. You're not supposed to be in here. Get out. So that's the difference. And it's striking. So not only does it happen right away, but the growth of the amount of antibodies once memory is achieved is nearly exponential. So it's definitely going to kick off quickly and hit hard to get rid of that infection. So for this reason, like, let's say this is chicken pox. Your first exposure, you're going to feel crappy. So if you get chicken pox, you're going to have an incubation period, and then you're going to start to get those lesions and get itchy and have fever and the other symptoms of that infection. If you get chicken pox a second time, the virus doesn't even have time to make you sick. The memory cells are so robust that they're just like, yeah, no, we're not doing this again. That sucked. And they bind up all of the new chickenpox virus right off the bat, disable it, don't let it get into our cells. And then that virus gets cleared from the system without ever feeling crappy or having any sort of symptoms whatsoever. So the first exposure can come in the form of infection that we actually fight off, or a vaccine. So in the case of a vaccine, the goal is to have the first exposure without actual illness. So you might feel a little crummy from a vaccine, and I'll explain why that would be in a minute, but you're not going to have full-blown symptoms of the actual pathogen because the pathogen is not alive. It's a vaccine that's teaching your immune system what the pathogen looks like without the pathogen actually doing anything to you. So the goal of a vaccine is to simulate a first exposure without illness, so that if you actually get exposed to the real, live, nasty version of whatever it is, you're able to do this, and then you never get sick to begin with. So that's the basis of vaccination, is create a situation where all of the steps I showed you just now happen, but they happen absent any illness. So I did mention, though, that sometimes people feel crappy for a little while after a vaccine, and that has had some very specific cultural consequences recently. So I think it's appropriate for me to address that a little bit more directly, because it would be good if everybody, there's some stuff about vaccines and how this works that is not common knowledge, and it should be, that helps explain this and make people not feel so scared. So that's what I'm going to do now. So let's talk about vaccines. So there's different kinds of vaccines. There's lots of possibilities for them. The very earliest ones were pretty gross. So I'm not going to tell like the whole long story, but the short version is there was a man who noticed that milkmaids did not get smallpox, and smallpox was awful. It killed so many people. And it's like chicken pox on steroids. Like, you get pustules, they're itchy, you get a fever, your throat closes, you die. It's very, very dangerous. It's a very good thing that we've eradicated smallpox using vaccines, actually. So this dude noticed that milkmaids didn't get smallpox and they didn't die from it. And he was like, wow, that's weird, because everybody else is dropping dead, but these milkmaids are fine. Why is that? So milkmaids get something called cowpox, which is similar to smallpox, but it only occurs on the hands and it goes away. And so he was like, I wonder if I take some of the goop from the cowpox sores on these milkmaids and I kind of stab it into another person, if I can give that other person the same immunity that the milkmaids seem to have against smallpox. And it turns out it worked. So that was essentially the first vaccination, was you take some infectious, gross pus from one person and you stab it into another person. And in doing so, you provide them with immunity. So it's called an inoculation. The reason that works is because cowpox and smallpox are viruses that are extremely closely related to each other. And so they look similar and the immune system can't really tell the difference. So it's just like, yeah, this is close enough, whatever, attack it. So vaccines can, nowadays, we don't have to use pus from milkmaid hands because that's disgusting. Nowadays, there's many different options. So we have this newer, it's not brand new though. mRNA vaccines have been around for around a decade. Everybody's like, oh, they're so new, I don't like that. I'm like, they're not actually, it's just that you haven't heard of them yet. So mRNA vaccines, which contain messenger RNA of viral genes, or sometimes bacterial components, because you can vaccinate against bacteria as well. Another option is so the option that's scary to most people is the live or attenuated virus and pathogen, because they don't like the idea that it's alive. What this means is it is a fully, like it's a virus with all the parts, or a pathogen with all of the parts, and that's good for recognition. So in that sense, it's quote unquote live, but it's been hobbled in some way. So the ability that it has to get into our cells has been removed, for example. So for COVID, that would look like taking the spike protein off. So people are afraid of these because they're like, well, why would you put a living virus in me if you're trying to not make me sick? But the part that they miss is that the virus has been molecularly disabled. So all it can do is float around and be noticed. It can't do anything. And there's also fragments or heat-killed pathogens. So you got some options, and which one is picked really depends on the pathogen in question. So all of these exist and they're all used for varying different purposes. But regardless, the point of them is they want to show the immune system what a pathogen looks like and cause production of memory cells. Here's the thing though, if you want to make memory cells, you have to do all the other steps. So you have to have recognition, sensitization, and activation of T cells, and you have to have the same for B cells. So you don't get to skip any of the steps that I just showed you for B cell activation and sensitization, and you also don't get to skip any of the steps for T cells. So because all of those steps, the steps that it takes to recognize, learn a pathogen, activate an immune response, make a bunch of cell copies, make a bunch of antibodies, all of those steps are coordinated by cytokines. So cytokines are a big, huge group of chemicals that white blood cells use to talk to each other, and they're also used to communicate to other cells and tissues. So turns out some of these make you feel crappy. So when people get vaccinated and they find themselves not feeling so hot, the conclusion that people that don't have a whole lot of biology knowledge, the conclusion that they sometimes draw is that the reason they feel sick is because the vaccine that they received must contain some accidentally there live pathogen, and that is making them sick, or that some component of the vaccine ingredients, a preservative. So what I was saying was the cytokines, some of these make you feel cruddy. And for some people, that causes them to draw the conclusion that it must be because the virus or pathogen was not killed completely, or that some component of the vaccine, like a preservative or like some of the stuff in vaccines that's used to make sure that they go where they need to go, maybe one of those things is bad or harmful or toxic. The word toxin is thrown around a lot in those discussions when it doesn't really mean anything in that context. People use the word toxin far too freely. So regardless, the observation that you sometimes feel crappy is true. Here's the thing though, it's not the vaccine component that's doing that. It's your own immune system. So for example, interleukin 6, and the interleukins, inter means between, and leuc means white. So interleukins are a bunch of proteins that white blood cells use to talk to each other. So interleukin 6, for one example, is not only used for communication by immune cells, but it also tends to cause myalgia, which is muscle aches. So the achiness that you might feel post-vaccine, not only at the vaccination site, but also elsewhere, that is sometimes caused by interleukin 6. The other thing to note, and I'm going to ask this Socratically, but if I don't get an answer because people don't want their voices on the recording, that's totally fine. I'll just wait and supply it myself if no one chimes in. But let's talk about humoral immunity specifically, because having a lot of antibodies is really handy when you're trying to prevent illness. So memory B cells would be the ones I'm referencing here. B stands for what?
Bone marrow?
Bingo. Thank you. Yes. So we talked in another recording about primary versus secondary lymphatic organs, and bone marrow is where the B cells first learn to do their job. But in addition to all of the B cells that already live in your lymphatic tissues, you have stem cells to make more B cells in your bone marrow. Now, as adults, we don't have that much red bone marrow left, but what we do have is primarily concentrated in our pelvis and the tops of our femurs. So in addition to the myalgia that interleukin 6 can cause, if you have been vaccinated recently and your immune system is mounting a response to the simulated infection that a vaccine provides, the amount of mitosis and differentiation of white blood cells is going to increase. And so in the bone marrow, for example, you would see a huge spike in cytokines. And in order to facilitate things like new B cells moving out of the bone marrow and into the blood, that includes things like inflammatory chemicals. So prostaglandins, histamines, and other eicosanoids that are associated with inflammation, and inflammation is painful. So the yucky feelings that people sometimes experience after a vaccination are actually a byproduct of the immune system's own activity in preparing and mounting an immune response. It's not any part of the vaccine that is causing damage to our cells or tissues. It's just the case that when you mount an immune response of any kind, whether it's to a vaccine or to an actual pathogen, it's uncomfortable. And part of the reason for that is our cytokines and the way that our cells signal and talk to each other. So the other thing was that was mentioned in the chat that I want to briefly address, although not too much, because I don't want to veer too far into politics corner, is just that people that are vaccine skeptical or nervous about them. I have a family member who is very nervous, who's not vaccinated because she's terrified. It has nothing to do with her political affiliation. She's just freaked out. And she is also in the same boat where she is deeply suspicious, partially because of the perception that the development was unusually fast. So here's what I will say about that, because it's another thing that, like, there's no reason for regular people to know this unless they're scientists. And so because that's not common knowledge, people draw weird conclusions about it. So the normal process of vaccine development is not happening during a pandemic. So it's typically, there's not as much of a rush. Not as many people are dying, although historically there have been some cases where it's been attempted before, like the earlier flus and some of the SARS viruses that we've had previously in the last like 20 years or so. But typically, if you want to make a vaccine, you have to do a bunch of research and development. And then once you have enough evidence that your vaccine idea is probably going to work, then you have to beg government for money to do further studies, clinical trials, animal experimentation that's federally required to make vaccines, et cetera. These things do not come cheap. And so scientists have to beg the government for money. So we have to beg all kinds of public health institutes to give us some of the tax dollars delegated to that thing so that we can do enough research to make the thing. And then after that, even more research, and then go back to step 2. So to get a drug or a vaccine from idea to FDA approved and implemented in the population, you have to go through these cycles. And research and development, specifically experimentation, take a lot of time and a lot of manpower. And the multiple cycles of having to beg the government for money and compete with other researchers for that money takes just so long. So that's the pace of science normally. However, the reason that the rollout for example, the mRNA vaccines against the original quote unquote variant of SARS-CoV-2 or COVID, the reason that rolled out much more quickly is not because the research and development was crappy or sloppy, but rather because the big hurdles of having to beg the government for money and have to compete with other research for that money, those were removed. Basically the government, the CDC, and other funding bodies agreed, we're going to put funding of other stuff on hold for right now because this is more important, and we need to make sure that we are making this process as smooth as possible for the people who are engaging in it. So it's not that it was rushed through, it was rather that the normal set of very serious and difficult to clear hurdles were just removed, which is great. And that I wish that would happen all the time, but it doesn't. It only happens in emergencies, like the one that we're currently living through. So that's the deal with that. Okay, so I think I'm going to stop recording there. Maybe. Can I, please?